In this last module on basic bioconductor infrastructure, I'd like to introduce two classes which come up a lot in bioconductor, which are the expression set and the summarized experiment. These are very similar classes of objects. The expression set is generally used for array-based experiments, where the rows are features, and the summarized experiment is generally used for sequencing-based experiments, where the rows are genomic ranges. So the expression set lives in the BioBase library, and I'm going to use an example from the Geo website. So there's a library GeoQuery, which lets you pull down expression sets by name. So if you have the GSC identifier, you can use that, provide that to the getGeo function, and this will download an expression set of microarray data. So we've downloaded this, and we've saved it to the GeoQ object, and this is a list which has a single element in it. And if we access that element, and let's save it to the letter E, just for simplicity, and in the top right we can see that E is a large expression set. It's about 10 megabytes. So expression sets are basically matrices with a lot of metadata around them. So if we just type it out, we see that we have an expression set which has assay data. This is the matrix I was referring to, which is 9,000 by 8. It has phenotypic data, and here we see the names and some different phenotypic information. It also has feature information, which we'll look at in more depth, and experiment and annotation information. So if you call dim, it gives the dimensions, which, like I said, was 9,000 by 8. You can also ask for the number of columns and the number of rows. The matrix, which contains the microarray values, are stored in the EXPRS slot, and you can access that with the EXPRS function. So if you want the first sample, you can use the column indexing. If you want the first row or the first feature, you can use the row indexing. You can also index these by name. So if we know the name of the first gene or the first feature, we can call it by name and likewise get this first row. The row names in this case are the probe sets, so already summarized into probe sets, which are genes, but it could also at an earlier stage be probes. So if we investigate the phenotypic data, we use the pdata function, and this gives us a data frame which is information about the columns of the expression set. So for each row here, this is corresponding to a column of the matrix. So these are information about the samples, including the accession number, the submission date, etc. And if we look at the names in this data frame, we can see we have the accession number, the status, and typically this characteristics underscore ch1 is the column which is used by Geo to describe the actual experimental state or condition. So here it looks like it's the strain information. And you can use the as numeric function on a factor, which will let you easily see how many replicates there are of each strain. So the dimensions of the P data, of the phenotypic data, should have the same number of rows as there are columns in the original expression set, so 8. And there's 31 columns of information about the samples. So the feature data is accessible with the fdata function, and this is information about the rows, which are genes or probe sets. And so this should have as many rows as there are rows of the expression set, which we see is true. And the names of the data in the feature data are for example, the gene title or the gene symbol or the Entrez gene ID or gene ontology information, which might be useful for downstream analysis. And so you can access these with a $, and you can see that there's 4,000 different gene symbols represented in these 9,000 rows. So it looks like there's some duplication of gene symbols. So I said there was also some experiment data and annotation data which is stored in this expression set. So in this case, the experimental data from GEO is blank, but it would give you the name and the laboratory. But at least we have the annotation for this expression set. So next I'm going to describe the summarized experiment object, and I'm going to load a bioconductor annotation package, which is the parathyroid summarized experiment library. And I'm going to load the gene-wise information, which is a summarized experiment, which summarizes counts of RNA sequencing reads in genes, in ensemble genes, for an experiment on human cell culture. I'll save this as a summarized experiment SE for convenience. And we can see that we have a summarized experiment object which with 63,000 rows, which are genes, and 27 columns, which are samples. And the matrix is, in this case, is called counts. And we have the row names, which are ensemble genes, and metadata about the row data and metadata about the column data, which I'll describe in a minute. So the dimensions of the summarized experiment is 63,000 by 27. And we can access these counts, which are the counts of RNA sequencing reads again, using the assay function. And so we see that the dimensions of this assay is indeed a matrix which has the same dimensions as the expression set. So col data, the column data, is equivalent to the P data on the expression set. So these are information. So each row in this data frame corresponds to a column in the summarized experiment. And we can see that there are indeed 27 rows here, which give information about the columns. So for example, the treatment, so each sample in this case is treated with 2 treatments or control, and we can see the number of replicates for each using the as numeric function again. So as I said, the rows in this case correspond to genes, and genes are collections of exons. So the rows of the summarized experiment is a G-ranges list, where each row corresponds to a G-ranges, which contains the exons which were used to count the RNA sequencing reads. So if we use the double square brackets, we extract out a single G-ranges object with 17 ranges and 2 metadata columns, which is the ensemble identifier for the exon, for example. So if we ask what's the class of the row data, we see that indeed it's a G-ranges list, and the length of this is then the number of genes. And if you ask what's the length of the first G-ranges, this will give you the number of exons for the first gene. So there's also included in the row data some metadata, which is accessible with the metadata function. And this is information which is stored, which is telling us about how this G-ranges list was constructed. So it was constructed from the genomic features package using a transcript database. It's a human, Homo sapiens was the organism, and the database was ensemble genes number 72, and etc. So that's a nice way to allow others to know exactly how this thing was constructed. In addition, there's some more information under experiment data using EXPT data, and then specifying the MIAME, which is minimal information about a microarray experiment. Although we're not using microarrays, we've still used the same slots to describe extra information about this object.